right, everyone. So I want to be really clear about exactly how we're going to go forward and use today as a test. So the goal is for you to have 3 different ways to sort of participate with lecture material prior to you taking that quiz that you're going to normally have as part of your weekly graded quizzes. Part number 1 is that you can watch short videos like this one and follow along with the slides. So I'm going to try and keep my video up so you can actually see me talk, and hopefully that'll help keep you interested. And we're going to keep them short, so they're going to be sections of those lecture slides. Part 2 is that you're going to do Socrative questions as you follow along. For those of you who had me for freshman year, you did this in my class, and we're going to try to mimic something that's pretty similar to that. If you haven't had me before, just rest assured, it's pretty easy. Basically, you'll open up your Socrative app on your phone while you watch this video, and then you just answer questions. I'm not going to do that today because I just want to make sure that the video is working for everybody. But I am going to be posting videos for tomorrow, and so you guys can follow along with Socrative as we're doing it tomorrow and make sure that that system all works. Part number 3 is what you're going to do after you watch all the videos, after you take your notes, and after you do the Socrative test, which, by the way, you'll just do once. It's something for you to be doing so that you're active during the videos. But once you've watched all of the videos for a particular set of lectures, there's going to be a quiz that opens up on Blackboard. These are going to be short 10 questions, but they're going to be, I'm trying to make them, randomized questions, which would mean that if you don't get the score that you want, meaning if you want an A in this class, you should get at least 9 out of 10. If you want a B in this class, you should get at least 8 out of 10, so on and so forth. If you don't get that score, it's telling you you need to work with the material more before you take that graded quiz. So it may mean that you need to watch another video. It may mean that you need to read your textbook. It may mean you need to rewrite or reorganize your notes. Or maybe you just need to come see me because remember, I will be on Zoom during class times. Whatever the case may be, those 3 things should be done. Watch the lectures, do Socrative questions as you work your way through the video, and take that Blackboard ungraded quiz, Web Campus ungraded quiz, all of those before you take your graded quiz for the week. Okay? Now, I'm going to keep posting more information and I'm going to keep being available on Zoom, but I just want to make sure everybody has a little bit of a start. So I'm going to work through the lecture material that we didn't finish before spring break so you guys can see how the videos are working. You might recall that we talked about DNA replication and that you guys drew the replication fork. Now would be a good time, if you haven't already done so, to try and test yourself to see if you can draw this replication fork again. I'm going to ask you guys to be able to work with the replication fork. Normally on exam 2, I would ask you to draw a replication fork from scratch. And unfortunately, you aren't going to be able to do that because of the nature of taking an online exam. But I am going to have a lot of questions about that replication fork that aren't just simply label the parts. A good way to make sure you understand how that works is to be able to draw it, because you can't draw it correctly if you don't understand how things work. The replication fork is something that's not specific to eukaryotes. The replication fork that you learned in your class is actually a prokaryotic replication fork. But the prokaryotic replication fork is something that looks a little bit different. All right. Now, when we look at the prokaryotic replication fork, what you're going to notice is that the nature of the fact that there's a circular prokaryotic genome means that there's a single replication bubble and that it works itself all the way around to the other side. That other side is going to be where these prokaryotes have to have a termination sequence. That termination sequence results in a nick being made in one side of that new strand, and then you can split into 2 new daughter DNA genomes. That nick gets healed by ligase. That's the same enzyme that puts ligates together Okazaki fragments. So in prokaryotes, it has an extra little spot that it helps. Remember that eukaryotes don't have to do this. As this eukaryote bubble opens up to these 2 directions, okay, these bubbles will simply merge here in the middle. Okay, so they can actually join. There you go. How's that for handwriting, guys? But what happens here at the end is very important. Because you're not interlocked with another circular genome, there's no need for a termination sequence, which means you don't need an enzyme called nickase to make that nick in one of the new daughter strands, which means you don't have to separate it and you don't have to ligate it back together again. These replication forks, as they grow, can simply merge together, and there's no need for that termination. Okay. Now, there we go. Oh no. Hold on. Let's get rid of my annotations. There we go. Okay. Now, all organisms, including bacteria, can add methyl groups onto their DNA genomes. So what we're talking about when we add a methyl group, if you look down here, you'll notice that there's this little M, which is attached to one of the cysteines. That's usually where methyl groups are added, is onto C bases. And it's actually literally a CH3, that little methyl group that gets added. But what it does is it slightly changes the shape of this base pairing. It doesn't stop C from base pairing to G, but the shape is slightly different. There are enzymes that bacteria had called restriction enzymes. So these restriction enzymes digest DNA at particular sequences. But those sequences are specific, and they're so specific that they notice the difference between a methylated cysteine and a non-methylated cysteine. So if your restriction enzyme cleaves DNA at the sequence ATTCGG, it will not cleave the sequence ATT methylated CGG. So bacteria can methylate their genomes, which will protect them from the restriction enzymes that they have inside of their cells. Those restriction enzymes will cleave other DNA, other unmethylated DNA. The reason that those restriction enzymes actually evolved is to cleave the DNA of infecting phage that gets injected inside of the cells. But we've also changed this methylation, or utilized this methylation in ourselves. So you'll find that humans have methylated genomes and others, even though we don't express restriction enzymes. So that's where this term epigenetics comes from. It's that idea that we can look at changes in genomes that are not based on the sequence of the genome itself, but a modification like this methylation sequence. This becomes very important when we talk about mutation, we talk about gene transfers. And if you guys choose to make, for example, a recombinant organism in your grant proposal that methylates DNA, or if you want to use restriction enzymes to help you make your plasmid, you're going to want to be aware of this. Okay. Now, as we talk about transcription, so this is what we're going to talk about for the next few minutes here. Understand that transcription is the process by which we make RNA. The enzyme that makes RNA is RNA polymerase, and there are different types of RNA polymerases that are responsible for making different types of RNA. In humans, in mammals, we have a different RNA polymerase for every type of RNA, but that's not really the case in prokaryotes that are a little more simple. The source of their RNA, the type of RNA that's made, really depends on the gene that's being transcribed. Regardless, there are 3 types of RNAs that are made. The first is a messenger RNA. That's the one that you know and love. That's the template for ribosomes to make proteins from. Ribosomal RNA is RNA that's never translated, but that RNA complexes with ribosomal proteins to make the actual ribosome itself. So ribosomal RNA is part of the material that makes the ribosome. But in order to make that RNA, you still have to have a ribosomal gene that you can use to actually transcribe to make it. Transfer RNA is going to be the RNA that's used during translation that actually provides that anti-codon match so that you put the right amino acid in. You'll notice that all 3 of these, in some way, shape, or form, participate in the process of translation, despite the fact that transcription is what makes those RNAs. So you might want to have an understanding not only of transcription, but how RNAs play a role in translation as well. So here's a question for yourself, and it's a fantastic example of where I would put in something like Socrative. But for now, I just want you to test yourself so you can see what it's like. Where does transcription start? And where does transcription end? This is something you should know from your previous classes. So you have options. Does transcription start at a promoter or a start codon? Hopefully, you said promoter. Promoters are binding sites for RNA polymerase. What binds to start codons? Initiator tRNAs. And initiator tRNAs are signals for ribosomes. Your small subunit binds. You have an activation of a GTP that provides energy, which recruits the large subunit, and that's what starts translation. If none of that's familiar, make sure you review before our videos that I post tomorrow. All right, so let's take a look at transcription in bacteria. RNA polymerase is assisted by what's known as a sigma factor. That sigma factor is like a transcription factor that you guys know and love. It helps to encourage this process of transcription. So you have to have a sigma factor. Bacteria use sigma factors, okay? And these bind to that core enzyme, that RNA polymerase, and to a promoter, so that it signals and it recruits RNA polymerase to the promoter. The same RNA polymerase transcribes all RNAs. So eukaryotes have a different RNA polymerase for every type of RNA. You have lots of RNAs being made all the time, and you have giant genomes. So why not make different specialized RNA polymerases? Bacteria don't have that. So they are going to have a single RNA polymerase that makes messenger, transfer, and ribosomal RNA. All of them are aided by a sigma factor, and all of them bind to promoter sequences at the start of the gene. Elongation factors are also going to be responsible for keeping RNA polymerase bound to the genome until it actually receives that termination signal, which is when RNA polymerase will fall off. Bacteria have different ways of terminating. Sometimes that termination signal is sufficient, and so the RNA polymerase simply falls off. But sometimes you have to have a termination helper, which is called Rho. So you'll see this term and hear about Rho-dependent termination. But that's something that bacteria use to aid them. So eukaryotes are going to be different in this process for a couple of ways. The first thing is that their transcription occurs in the nucleus. Remember that there is no nuclei in prokaryotic cells and there's no division. So the genome is hanging out in what is essentially the cytoplasmic region. Yes, it's condensed in a nucleoid, but it's still part of the major cell. And so that's going to be the source of, and the location in which transcription occurs is the quote cytoplasm. Eukaryotes have a DNA genome hidden in their nucleus, so transcription is going to occur there. They also have 3 major polymerases for the 3 major RNA types. And they're going to be using transcription factors like this sigma factor. I'm going to draw you back here to the idea that all bacteria use a single transcription factor called a sigma factor, but eukaryotes are going to have lots of different transcription factors. Many different ones. I can't possibly teach you them all because every gene not only has a single transcription factor, but most of them have many. So take a look at the example on your slide. Here's a eukaryotic transcription factor complex. There are a lot of transcription factors that are responsible for this here and that are recruiting, in this case, RNA polymerase 2. Okay, so those are all going to be binding to a promoter, just like sigma factor binds to a promoter. So the function is the same, but they've evolved a lot of different versions. So bacteria use that sigma factor, and archaea use what's known as transcription factor B. So transcription factor B is going to recruit RNA polymerase in a very similar fashion to the way that eukaryotes use it, but archaea just still haven't evolved that diversity that eukaryotes have. So much like the other attributes of archaea that we've talked about as contrasts, so when you guys were specifying and talking about what makes archaea different from prokaryotes or eukaryotes, this is actually another area of contrast. Even though we have the simplicity of a prokaryotic cell and that lack of diversity, it functions similarly to a eukaryote. So the transcription factors involved in this process are a little bit different. Eukaryotic and archaeal promoters often include a TATA box. So you guys probably have heard about this in genetics. The sequence is shown on the slide, TATAAA. That's a TATA box. That's why we call it a TATA box, because if you were to read it out, that's how it would sound. And if you see that TATA box, you know that you're dealing with either a eukaryotic promoter or a archaeal promoter. Prokaryotic cells, bacterial cells specifically, are much more likely to have what's known as a Pribnow box. So it's a TAT-TAT. It's not a big difference, but you'll note that that last A gets changed to a T. So bacteria have this special Pribnow box. If you ever receive a sequence, you can look upstream, so to the left before the start codon, and you can look for one of these two sequences. If you see a TA, TAAA, you know you're dealing with either an archaeal or a eukaryotic protein. If you see TA, TAAT, that's called a Pribnow box, and it indicates to you that it's a bacterial protein. So these promoter sequences that specifically bind transcription factors, they're going to help you to figure out exactly who that sequence belongs to. Eukaryotes also have elongation factors and termination factors. So remember that in bacteria we had this idea of this RNA promoter or this RNA polymerase could kind of fall off. Well, it will either fall off on its own or it will fall off in a row-dependent manner. So Rho is actually going to be a protein that sits there on the termination factor or termination sequence and helps to signal RNA polymerase to fall off. So that's what bacteria use. Eukaryotes have poly-A tails, and so these poly-A tails and this poly-A signal sequence has an added level of complexity. So if you're looking at, for example, here, so here's our RNA transcript that is released. If you have at the end here this poly-U sequence, there's a structure that's actually being created. This Rho termination protein is going to act a little bit differently in that it actually moves its way up through and disconnects the RNA polymerase. So I don't want you to know huge amounts of detail about this, but I do want you to understand that there are different ways in which we can terminate. If you are a eukaryotic cell, the termination of your transcription is going to follow a poly-A tail. So you're going to have a signal sequence which indicates that you are going to have that traditional 3' poly-A tail. Okay? So there's a poly-A sequence. That doesn't happen in bacteria. Bacteria don't modify their RNA, coming up in a second. Because they don't modify their RNA, they're not going to use a poly-A tail. They can either create a structure at the 3' end of the transcript, shown here on the left, which causes the release of that RNA polymerase, or they can use a protein called Rho, which physically disrupts the RNA polymerase and causes it to fall off. Clearly, these are different than having a poly-A tail. So let's talk about the idea that eukaryotes can do this RNA modification. So you all know the poly-A tail shown here on this 3' end, and hopefully you know that at the 5' end there's a cap which is attached to the end. It's actually a modified guanine with all 3 of these phosphates attached to it. So you can't attach onto that 5' end, but it's clearly a different shape, and it helps to signal the 5' end of that transcript. So these RNAs in eukaryotes, okay, within this nucleus, so before they leave the nucleus, they get capped with a 5' cap, and they get a 3' poly-A tail. This doesn't happen in bacteria because prior to the end of transcription, translation already becomes available. Remember, ribosomes are found in the cytoplasm. If you are making an RNA transcript in the cytoplasm, that means they're already exposed to ribosomes before you're even done. This is going to be really important when we talk about translation more next time. The other thing that occurs in eukaryotes is splicing. So you might remember this idea that there are spliceosomes. They're actually RNA, called small nuclear RNAs, that participate in this process. And they're capable of cutting exons out of eukaryotic RNAs. So you can get alternative splicing. You can get one gene that results in multiple different types of mixed-matched proteins simply because of the fact you've separated the process of transcription and translation. You can't do that in prokaryotes. So we're going to talk next time about operons, because operons allow for some of that diversity that prokaryotes need to have. Eukaryotes, they can achieve diversity in different ways, such as RNA processing. I want you guys to make sure that you can go through this chart and complete it from start to finish. Now, I haven't finished all of it for you, but you're going to want to be able to compare these. So as you guys are working through information, it's important to not just create a linear set of notes, but to actually organize that information for yourselves. It will make it a lot easier to remember. Okay, next class is going to be a busy one, particularly because there's going to be 3 short videos like this so that you can, at least I think it will be 3, pay attention, at least 3 short videos like this so that you can kind of see how this stuff is going to work. And I'm going to add to them those Socrative questions so you can follow along. For the time being, hopefully you guys got through this video okay. If you have any concerns about the videos and being able to see them, please don't hesitate to reach out so I have some time to adjust them before I post the others. And let me know if you have any questions or if anything worked great. Alrighty, I will see you guys tomorrow.